All right, welcome to INBDE Microbiology review series number 2. Previously, we had review series number 1, where we talked about the difference in size of bacterial and eukaryotic cells, and we discussed about the DNA in the bacteria, where it's found. We say that it's found. DNA in bacteria is found in the cytoplasm. Okay, and the DNA in eukaryotes is found in nucleus. All right, and we also discussed that because the DNA in bacteria is found in the cytoplasm, the transcription and translation are coupled processes. Okay, however, in eukaryotes, the transcription and translation are not coupled. All right, so those are the things you have to keep in mind. Make sure you review those if you haven't reviewed it yet. Okay. All right, so we're going to talk about the peptoglycan, okay, which make up the cell wall of bacteria. Okay, so peptoglycan is a linear polysaccharide. Okay, it's a linear polysaccharide chain which is cross-linked by short peptide chains. Remember, it's pepto, which means protein. Okay, has peptine, protein, and then glycan, so meaning it has some sugars, some carbohydrates, so saccharides. Okay. All right, so this is very important. So the cell wall is made up of a molecule called peptoglycan, and it is made up of protein and some carbohydrates, which is saccharides. Okay. So the polysaccharide chain has repeating disaccharide, okay, of N-acetylmuramic acid, or AIM, and N-acetylglucosamine as the repeating disaccharides. Very important point. Okay, so I'm going to repeat it. The polysaccharide chain in the peptoglycan is made up of repeating disaccharides. Okay, a disaccharide which is made out of N-acetylmuramic acid and N-acetylglucosamine. The polysaccharides are cleaved by host lysozymes, okay, yielding protoplast and spheroplast, which lies unless there's osmotically stabilized. Okay, unless they're osmotically stabilized, they are not lysed, so they become stable. Okay. All right, but you have to remember that the disaccharides which make up of the peptoglycan are repeats of N-acetylmuramic acid and N-acetylglucosamine. The cross-linking peptides in a peptoglycan, they vary in species, meaning different types, but they are always linked to carbohydrate chain via the muramic acid. So they are not linked. The peptides are not linked to the carbohydrate by the N-acetylglucosamine. They are linked by N-acetylmuramic acid. Okay, very important point. Okay, that you need to remember. All right. So the cytoplasmic membrane has membrane-bound transpeptidase. These are enzymes which characterize the cross-linking reactions that join peptides together. Okay. And these peptides, okay, transpeptidase, are called, all right, penicillin binding proteins. So initially, when penicillin was discovered, it was found to bind to these proteins, which are found. So these penicillin binding proteins are found on cell membrane, and they act as enzymes uh, called transpeptidase. They join the peptides to form the peptidoglycan. Okay, and so they are called penicillin binding proteins because they are targets for penicillin and other beta lactam antibiotics. Okay, so you cannot review peptidoglycan without knowing the penicillin binding proteins. You have to know those penicillin binding proteins because these are common sites for the antibiotics such as penicillins and the other beta lactam antibiotics. Okay, all right. So the during infection, peptidoglycan can interfere with phagocytosis. Remember that that's the one of the importance of the cell wall of bacteria. They use it to prevent the macrophages or dendritic cells from phagocytosis. Okay, so cell wall is critical for the bacterial survival. All right, or evasion of the host immunity, and so it also stimulates mitosis of lymphocytes, okay, which helps in the immune system and induce fever, okay. So fever is called pyrogenic activity, all right, when you're producing fever. So the cell wall itself, it helps the bacteria to evade the host. However, when the host sees that cell wall, also it induces the production of more lymphocytes, of more immune cells, okay. And this is critical in terms of vaccination. So you can take out the contents of bacteria and have that cell wall and still cause the uh, 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 vaccine effect in the host. Okay. So now you have to know that the cell wall in the cell wall, the peptidoglycan, okay, is mostly found in the gram positive bacteria. So gram positive bacteria have a thick peptidoglycan. Gram negatives, they do have it, but it's very thin. Okay. So gram positive bacteria have thick peptidoglycan wall, which traps the gram stain. Okay. Gram stain is a crystal violet iodine complex. Okay. However, the gram negative bacteria have a thin peptidoglycan cell wall, and this allows the stain to be readily washed away. So it goes in, and then when you wash it, it washes out. So you don't see the gram stain anymore. Okay. So you have to know the differences in terms of the structure of the gram positive and gram negative bacteria. So the exceptions on the gram stains, okay, you have mycoplasma are gram negative because they lack peptidoglycan layer altogether. Okay, although the mycobacterium have a typical gram positive cell wall, their waxy outer shell of mycolic acid. All right, waxy outer shell of mycolic acid prevents the penetration of the stain, so they appear as gram negative. All right, even though they have a thick peptidoglycan layer. Okay, so mycobacteria are identified using acid fast stain. These are critical things that you need to know. Okay, all right. All right, so our next one is going to be the review series. So make sure you review series number 3. Make sure you review series number 1 and number 2. Okay. All right.